গৰম উঠিছে ন দি দিয়া অকণমান দেৰি অলপ পিছত বন্ধ কৰি দিবা 5 মিনিট দি দিয়া অ'কে লাষ্ট ক্লাছ কি কৰাইছিলোঁ লাষ্ট পইণ্ট কি লিখিছোঁ হা পেপটিডোগ্লাইকেন আজি টিকইক এচিড লাষ্ট ক্লাছ কৰিছোঁ অ'কে চ' আমি লাষ্ট ক্লাছত কি ডিছকাছ কৰিছিলোঁ উই ৱেৰ টকিং এবাউট the kingdom called as Monera or tare bacteria in details we were discussing. We were talking about the cell envelope, right? Tinta layer, right? Bacterial cell wall is made up of key peptidoglycan and we will be writing about that. Reading Cora, peptidoglycan. Point number 2, no? 4. So peptidoglycan of ki mon rakhiba cell wall of bacteria is made up of ki peptidoglycan. Excuse me. Excuse me, I'm not well. তোমালোকৰ লগৰ যে আৰু এটা ল'ৰা আছিল কি নাম আছিলে তাৰ কি এৰিলে নেকি বায়ো নেকি ক্লাছ কৰা নাই Whole. Okay, so peptidoglycan, it makes the cell wall. Cell wall of bacteria is made up of peptidoglycan. Actually, peptidoglycan is made up of key. It is made up of amino acids, right? So different amino acids in the peptide part, in the glycan part. Iman mukhasta koribo nalage. What you have to remember is this is the first slide. Then second is আমি gram positive bacteria or gram negative bacteria কিছুমান unique points পঢ়িব লাগিব ন you have to differentiate between gram positive and negative positive or যোৱা ক্লাছত if you remember teichoic acid বুলি এটা word পঢ়িছিলো teichoic acid is the characteristic of gram positive নে gram negative gram positive ঠিক তেনেকে ইয়াৰ question আহিব DEPA is a characteristic of gram positive নে gram negative DEPA is a characteristic of gram negative this is the only word that you will remember out of everything is the point clear baki bilakor upor question aha nai gram positive or eta beleg amino thake gram negative or eta beleg thake so teichoic acid is a characteristic of gram positive DEPA is a characteristic of gram negative right and next is you also have to remember neg and nem because no doubt say almost all bacteria neg and nem thake kintu ami pisot dekha pam there are certain type of bacteria jot diki na thake so as of now just remember this word this line and these two words that's all so this is all about the cell envelope it is next part of bacteria now ami ki discuss kori asile prokaryotic cell and its structures next likha staining in bacteria So in the last class we have spoken a little bit about this process, right? Staining korar basis hote ki hoy gram positive or gram negative ami differentiate koribo paru, right? And first is why do you stain bacteria? To study the contents better, right? Microscope tolot jate bhalke dekha pao. So what is important is staining process discovered by or developed by and what is the stain that is used, right? So pore ami different stains porim cell to ami jeta porim na we have different organelles stained by different colors, right? So question I have bacterial stain or naam, mitochondrial stain or naam, etc etc. So we are talking about a bacterial stain of ki boli ko crystal violet boli ko. Next likha technique বা steps বা procedure কি likha. So gram staining process to এখন flow chart or form di dim. Write it down. So what we are writing is the process of staining a bacteria, but in the form of a flow chart instead of writing sentences. যোনখিনি নহয় তাক আকৌ ষ্টেইন কৰিব লাগিব ভাইলেট ওলালে যদি পাৰ্পল বা ভাইলেট ওলালে তেতিয়াহ'লে তাহাঁতক গ্ৰাম পজিটিভ বেক্টেৰিয়া ক'ম আৰু কিছুমান বগা হৈ থাকিব যোনখিনি কালাৰখিনি ৰিটেইন নকৰে ন এতিয়া মই বুজাই দিম এতিয়া লিখা হ'ল ন লিখি লোৱা No? So what we are doing is we are writing the techniques the bacteria tak stain kenege korim and on the basis of stain we differentiate between the gram positive and gram negative, right? The first question is what is the stain that we are using? So bacterial stain boli kole we understand that we talk about crystal violet, which is also called as gentian violet, which is also called as gram stain, right? Eketai naam. So what you do is bacteria lua bacteria color kora color dia pisot ki hobo first duta part ma olop pisot bujam. So you stain the bacteria gets stained, I think it's understood. Then what you do is you go for washing, right? Stain to deeply wash kora pisot ako ki kora you restain, right? So bacterial staining ot ami stain keta use koru, bacterial staining ot stain ami duta use koru. No doubt we will be talking about this stain also kintu ekhini pisot this is the main step. So main step ot ami stain keta use korilu duta, so hetu upor question ahe কি stain crystal violet or iodine যেহেতু দুটা stain দিলো দুইটাই মিলাই পেলাই এটা কালার ফর্ম কৰিব ন আগতে কি দিছিল crystal violet দিছিল তাছ আকো কি হেৰে stain কৰিছে iodine সো দুইটাৰে কালার থাকিব সো দুইটা কালাৰক আমি কি বুলি কৈছো CVI complex, right? Crystal violet iodine complex, so bacteria would appear like that, right? After that process what is the next step? You do a treatment with alcohol. Treatment মানে কি আকো you wash it ইয়াতো এবাৰ ওয়াশ কৰিছে ইয়াতো এবাৰ ওয়াশ কৰিছে সো কোৱেশ্চন আহে গ্ৰাম ষ্টেইন প্ৰসেছটোত ওয়াশিং কেইবাৰ হয় ওয়াশিং দুবাৰ হয় ৰাইট ফাৰ্স্ট টাইম কি হেৰে হৈছে ওয়াটাৰৰ সেকেন্ড টাইম কি হেৰে হৈছে এলকহলৰ হৈছে আফটাৰ দিস প্ৰসেছ ন once you wash the bacteria with alcohol some of the bacteria retain the stain colorful হৈ থাকিব ধোৱাৰ পিছতো আৰু কিছুমান কি হ'ব de-stain তাৰমানে they will not retain the stain. Do you understand between retaining stain and do not retain stain? Yata eta question ase june retain kore tak ami ki boli kom gram positive ase color, so they will obviously be appearing purple and violet, right? Junkhini color de-stain hol eta de-stained mane ki they will not take the color and they will not retain the color. There are two things, no? So a negative bacteria khine color to crystal violet lobo ne noloy retain koribo ne nokore, so tahat luat to loboi কিন্তু retain হে নকৰে so gram negative bacteria they will take up crystal violet but they cannot retain it. Gram positive bacteria they will take the color and then they will retain it. So যেহেতু retain কৰিলে we got the gram positive, right? Rest of the bacteria that could not retain the stain তাহাঁতকো পঢ়িবলৈ হলে you have to use another stain that is called as safranin, which is pink in color. And because of this color bacteria কি হয় pink হৈ যায়, right? So yete a question up till here যে কোনটো স্টেপে gram positive আৰু negative ক differentiate কৰে. We have written so many steps, no? কোনটো স্টেপে, right? So alcohol wash কৰাৰ পিছতহে আমি গম পাম তুমি gram positive আৰু gram negative. I think the process is understood. So from both point of view also যে লিখিবলৈ you can write it in sentences. Examination I mean entrance point of view পৰা sentence দিয়ে the points that we have marked. Washing কেইবাৰ হৈছে, color কেইবাৰ use কৰিছে, কোনটো ষ্টেপৰ কাৰণে you differentiate, etc. etc. Now talking about this entire process, we have started with something called as bacterial smear and air drying. But আমি কি জানো যে bacteria to color কৰিম, bacteria color কৰিম, instead of writing bacteria what we have written bacterial smear. Now what do you understand by the word smear or smearing? Smearing আমি তো bacteria বুলি লিখা নাই না, bacterial smear বুলি লিখিছো কিহৰ কাৰণে আমাৰ what is the aim? We have to stain the bacteria, but we know you cannot be collecting a single bacteria with your hand. এটা এটাকে ব্যাকটেরিয়া ধৰি আনিলো তাক ৰঙত ডুবাই দিলো তেনেকুৱাটো নহয় ন. সো জেনাৰেলি ব্যাকটেরিয়া ক'ত থাকিব, পানীত থাকিব. সো what you will do is you will collect a drop of water, এখন গ্লাস স্লাইড ল'বা, তাত ড্ৰপ অফ ৱাটাৰটো দিবা আৰু এখন গ্লাস স্লাইড দি what you do is slide it down. যিখিনি বস্তু লাগি ধৰিলে that process would be called as smearing আৰু সেই যিখিনি লাগি ধৰিলে that would be called as smear, right? আৰু সেই যেতিয়া এখনে ঘঁহি দিলা তাত ব্যাকটেৰিয়াখিনি লাগিলে তাতো এক্সট্ৰা ৱাটাৰো থাকিলে ন, সেই এক্সট্ৰা ৱাটাৰখিনি আমি কি কৰোঁ air dry. Is the point understood now? Bacterial smear, right? So we collect the bacteria, smear to law, and this is the process, right? So this is all about staining. Next likha classification of bacteria. So chart format nilikiba, extra ke heading koriba, right? So classification dukhon korim on the basis of shapes of bacteria and on the basis of flagella, right? Okay, so we are talking about the shapes of bacteria. We have written this part. A to part iman important no. What is important is whenever we talk about shapes, there are 4 basic shapes, and what are the basic shapes, right? Coccus, Bacillus, Vibrio, or spiral. And whenever we use the word coccus, we need to understand that the bacteria is spherical, right? Bacillus will be called a rod shaped, and we understand these 2 terms, right? Apart from those 4 basic shapes, কিছুমান স্টকড বেক্টেৰিয়াও থাকে স্টকড বেক্টেৰিয়া মানে কি এটা বেক্টেৰিয়া এডাল ষ্টক থাকিব খাৰী এডালৰ নিচিনা থাকিব right and we have budding bacteria also especially when we talk about budding is something that goes very well with yeast right yeast কি কৰে budding কৰে right so budding মানে কি নিজৰ বডিটোৰ পৰা এটা পাৰ্ট বাডৰ নিচিনাকৈ ওলাই আহি পেলাই সেই পাৰ্টটোৱে আকৌ নতুন ইন্ডিভিজুৱেল হিচাপে it will behave right we have a term that is called as pleuromorphic bacteria morphic mane morphological shapes pleuro mane many normally when we talk about bacteria specific shapes thake no coccus jodi coccus bacillus jodi bacillus etc kisman bacteria different shapes thake that is what is called as pleuro many morphological shapes pleuromorphic bacteria eta example rhizobium leguminosarum rhizobium pisot malge poriba have you heard something called as nitrogen fixation nitrogen fixation শুনিছা ন soil fertility বঢ়াবলৈ হলে nitrogen fixation কৰে right nitrogen fixation কোনে কৰে microorganisms এ কৰে right কিহৰ কাৰণে কৰে so that the plants can take in nitrogen right nitrogen কৰ পৰা লয় first the plants of nitrogen লাগে না লাগে obviously লাগে right কিয় লাগে because basic amino acids proteins form কৰিবলৈ nitrogen লাগে নাইট্ৰজেনৰ সোর্স কোড যেনেকৈ প্লান্টস বিলাকে অক্সিজেনও লাগে প্লান্টস বিলাকক কাৰ্বন ডাই অক্সাইডও লাগে কাৰ্বন ডাই অক্সাইড কৰ পৰা লয় atmosphere পৰা লয় অক্সিজেনও লাগে ৰেসপিৰেশনৰ অক্সিজেন কৰ পৰা লয় atmosphere পৰা লয় প্লান্টস অফ নাইট্ৰজেনও লাগে নাইট্ৰজেনৰ সোর্স কি নাইট্ৰজেনৰ সোর্সও একচুয়ালি atmosphere right কাৰণ নাইট্ৰজেনটো সইলত নহয় না নাইট্ৰজেন অৰিজিনালি সোর্স কোড আছে ইটস ইন atmosphere কিন্তু atmospheric নাইট্ৰজেনটো প্লান্টস বিলাকে ডাইৰেক্টলি ল'ব নোৱাৰে একাৰণে atmosphere পৰা আহি পেলাই সইলত মিক্স হ'ব লাগিব that is what is called as fixing right and the best example could be rhizobium so okay coming to this etia ki poribo lagibo ihotor classification right heading likhiba coccus ekhini first moi likhi no tarpsot we like so when we talk about coccus the first word that we understand is je shape to keniko round right keniko types of coccus thake coccus bacteria bahut type of classification ase jeneke if I use a word called as monococcus ki buji paba jodi enuka words bilak use koru monococcus diplococcus tetracoccus setia hale collection of one collection of 2 collection of 4 right so accordingly we have classification of coccus classification of bacillus etc So caucus or classification, kiman types? 6 types, right? 6 types or words bilak to I think understanding is easy. Monococcus, diplococcus, it's easy. The good part is that we don't have to remember the examples. No doubt we have written the examples, but examples don't read. What is important is maybe if the question is asked on the words that we have underlined and maybe the diagram. If you have a diagram to read, you have to identify monococcus and diplococcus. Looking at the diagram itself, you will understand. At least monococcus, diplococcus, tetracoccus, right? We have a word called as streptococcus. Streptococcus, strepto mane in the form of a chain, right? So what you need to understand is that meaning of that word. Strepto mane chain. Etia moi jodi streptococcus no goi pelai streptobacillus ko. Means what? All the bacilli bacteria arranged in the form of a chain is called as streptobacillus, right? So question hai strepto, a bacteria are arranged in the form of a chain, it is called as. So the word that is important is chains, right? Yat jane ke okole ase no, yate ki hoise chain or nisa ke ekelage lagi ase. So chain mane strepto mane in the form of a chain, right? Again what is important is the meaning of stephilo. Stephilo mane irregular bunch. Bunch buzi pao no, bunch of grapes, right? Jeneke yate what group ase, yate kita specifically sarita ase. Hekane tak ami ki boli koisu tetra. Yate eko specific nai. So it is called as ki irregular. So what are the points that is important? The word stain and strepto, right? What is important is irregular and stephilo, right? Next another that is important is Sericini. Jedi bacteria bor eneke arrange hoi thake that it looks like a cube of 8. That combination would be called as ki Sericini. Obviously examples not important, the meaning of the words are important and the diagrams. Clear? So tate kobat likhi ethwa examples not important karon pisole pohibo na lage ni basically pohi to lab nai no. Examples er upor etu part of question aha nai. If this is over bacillus heading kora. Yar ekita word buzi palu? Oh, another word that's very important is we have marked a word called as aflagellate, right? Aflagellate mane flagella nai, right? Normally bacteria flagella asa nai. Remember the diagram that we have drawn in the first class, right? So bacteria ki hoy, they are motile, tahandor flagella ase, kintu coccus bacteria bilakor flagella na thake. So heitu upor to question ahe, flagellated ne aflagellated, right? Next लिखा bacillus. Bacillus or shape केनेকুৱা আছে না rod shape. Okay, coccus কি আছে flagellated নে aflagellated? Aflagellated. Bacillus কি হয়? Flagellated নে aflagellated? Flagellated আৰু generally flagellated ৰ মাজত difference কি? Exceptions আছে. So ইয়াৰে এটা exception আছে. Right? Next is bacillus. Bacillus shape to lagibo rod shape, generally flagellated, or most common shape out of the 4 shapes, right? Next is kiman type, 4 types. Coccus kiman asile 6 types. Mono, diplo, strepto. We understood mono single, diplo double, strepto ki asile in the form of a chain, right? All the bacillus bacteria would be appearing or would be arranged in the form of a chain. In this part, we have something called as pellicid. Tat jeneke ami Sercini porisilu, right? Ketaman word poriso yat pellicid. Pellicid, or what is the word that you need to remember is matchstick, right? Arranged in the form of a matchstick. Edal dal ke matchstick jodi arrange karo, that arrangement would be called as pellicid, right? So question has been asked on this word, and etu word ahile you have to mark this one. That's all. The third shape is vibrio. Vibrio eko details nai. Mane nalage tumaloko. So আমি কি কৰি আছো shapes of bacteria য'ত লিখিছে examples not important বুলি লিখি থবা কোনো এটাৰ example পঢ়িব নালাগে right মানে এনেকে বেলেগ ফৰ্মত পঢ়িব লাগিব লাইক কিন্তু এনেকে নাহে give an example of a monobacillus bacteria তেনেকে নাহে বা এইটোও নাহে যে lactobacillus is an example of তেনেকে নাহে example or নাই আহা diagram and the specific words that are important. So we are talking about classification. First classification on the basis of shapes, second classification on the basis of flagella, right? So next likhiba number of flagella. এখিনি হৈছিলে So classification part or maximum asked question is obviously from this part, right? Number of flagella. Apo examples put important no hoy. What is important? The terms, the diagrams, right? And obviously the words, the sentences that we have written. For example, side of diagram kita kita baat nahi. Clostridium tetani. Dium tetani causes tetanus. Our diagram kita sound sentence kita buji paiso, right? So sentence kita di dibo, terms kita kudibo, but terms kita di dibo, you have to know the sentence. For example, the question could be a bacteria which has flagella all over the body is called as, it is called as peritrichus, right? Which has one or more flagella arising from both the ends, it's called as lophotrichus. So from one end, from both ends. I think looking at the diagram we understood. So what is important? Side or likhi over the terms, that's already tomage tick kori side. Examples not important. And out of this, maximum question has been asked on peritrichus. Next, because structure of flagella. So when we talk about the flagella of bacteria, it can be divided into 3 parts. Right? You have the basal body, the hook and the filament. Likha hoise? So when you talk about the basal body talks about what? Base of the flagella. Base mane ki? Jot lagi ase. If you remember the diagram that we have drawn in the first class, no? Eneke diagram eta akisilu no? Tade tume hair like structures disilu ar flagella dal ekhini pura disilu no? So flagella ta kor pura disilu? E bahiror etu layer or pura ne bhitoror pura? Obviously bhitoror pura, right? Bahiror layer to ki boli koisile? Capsule. Right? So capsule pura originate kore niki? Nokore. So tar flagella koloke lagi thake base of the flagella to some extent cell wall and cell membrane, right? So cell wall or cell membrane or ekini hol base, gutekini basal body to cell wall or eta portion lagi ase, cell membrane or eta portion lagi ase with certain ring like structures. However, this part of information is not important. Why it's not important? Because NCERT book iman khini details diya nai, right? NCERT kunkhini pura dise, ekhini pura dise. So flagella of bacteria is made up of 3 parts: the basal body, hook and the filament. It looks something like this, right? Information has been given. Out of all the information from entrance point of view, what is important is this one, right? So question kiya hai, so whenever we talk about flagella okol ki prokaryotes ore ase ne eukaryotic cell kisumanor ase, eukaryotic cell oru ase, kintu the eukaryotic flagella or prokaryotic flagella differs, right, in the protein structure that they have. Eukaryotic flagella is made up of tubulin protein, or prokaryotic flagella is made up of flagellin protein. So entrance point of view pra he point to important, right? Bacteria ki hoy, eukaryotic ne prokaryotic. So prokaryotic flagella is made up of flagellin protein, right? Or as far as this is concerned, LPSM ami okman extra likho, yate aru eta point likhim pisot etu kori do ekin loke. লিখিছো পইণ্ট কিন্তু নট ইম্পৰ্টেণ্ট, মই মাৰ্ক কৰা নাই। Excuse me. Right? আৰু এই যে চাৰিটা ৰিঙৰ কথা কৈছে, outer pair of rings আৰু inner pair of rings. Outer pair আৰু inner pair refers to what? The cell wall and the cell membrane. ভিতৰলে সবতছে ভিতৰলে কি আছে? cell membrane খন. So তাত থকা ring কিটাক আমি কি বুলি কম? inner pair. আৰু cell wallৰ লগত লগ লাগি থকা ring কিটাক আমি কি বুলি কম? outer pair attached to the cell wall. এতিয়া sorry, এটা হেৰি কৰাচোন, হেডিং লিখা differences between gram positive and gram negative. Differences between gram positive and gram negative কৰা chart এখন লিখা, এপিনে gram positive, এপিনে gram negative লিখা। So হেডিং কি লিখিছা ডিফাৰেন্সেছ বিটুইন গ্ৰাম পজিটিভ এণ্ড গ্ৰাম নেগেটিভ ফাৰ্ষ্ট পইণ্ট লিখা লিখাচোন গ্ৰাম পজিটিভত লিখা ৰিটেইন the blue color of gram stain retain the blue color of gram stain even after washing with alcohol retain the blue color blue color of gram stain even after washing with alcohol ipine they get destained second point a cell wall thick or epine thin cell wall thick thin gram positive or thick aru gram negative or cell wall boli likha point number 2 cell wall likha dash edal diga thick boli epine likha epine thin boli likha next point gram positive it is not covered by liposaccharide LIPOSACCCHARIDE not covered by liposaccharide layer that is jano no I dot E etu jano ne that is the cell wall is single layered ipine likha it is covered by liposaccharide layer so the cell wall is double layered it is covered by liposaccharide layer so the cell wall is double layered etu kuntu point er upor likhisu ba so the cell wall is double layered ami eta diagram akisilu no peptidoglycan eneke di pelai right teichoic acid disile epine ki disile gram negative or akko eta layer asile nai yate kintu nai so this is the point that we are writing here right okay next point number 4 no gram positive or it is covered by liposaccharide layer so the cell wall is double layered next point more rigid RIZID due to high concentration of peptidoglycan tar mane peptidoglycan beshi ase due to high concentration of peptidoglycan are epine less rigid next point muramic acid boli eta acid ase muramic acid that makes up peptidoglycan thik ase positive or likha muramic acid 70 to 95% yat likhiba 5 to 20% 5 to 20%. Next point. Lipid content is low. Yate high. Next point. Teichoic acid present. Teichoic or? Teichoic acid is present, teichoic acid is absent. Next point.
Negatively absent.
Teichoic acid অকল gram positive থাকে, right? Next point. Remember this? অলপ আগতে কৰিছিলো, right? DAPA absent in gram positive, present in gram negative. DAPA কি হয়? Amino acid, right? Present in gram negative. Next point. Gram positive or likha. The cell wall is more sensitive. The cell wall is more sensitive to antibiotics like penicillin. Antibiotics, antibiotics, antibiotics like penicillin. Not so sensitive to penicillin. Not so sensitive to penicillin. Gram negative what? All? Not so sensitive to penicillin. Next, gram positive. Mostly capsulated. Capsulated jano no? Presence of capsule. Mostly capsulated. Sorry, mostly non-capsulated likha. Gram positive what? Mostly non-capsulated or gram negative what? Mostly capsulated. Last class or ami jete capsule পঢ়াইছিলো, what did we say? যে যার capsule খন থাকে, first of গুটে bacteria সব bacteria capsule না থাকে, কিছুমানৰ থাকে আৰু কিছুমানৰ instead of a capsule what you see is the presence of a slime layer, right? যার capsule থাকে তার কি হয়? protection বেশি থাকে, আর protection বেশি থাকে এখানে you cannot kill them easily, and plus আৰু এটা character that we have read about capsulated, virulent one to bond আছে নে তাহাত কি হয়? disease causing. so gram positive আৰু gram negative compare কৰিলে disease causing maximum কি হয়? gram negative হয়, এখানে capsulated কাৰ হ'ব? gram negative ৰ, right? so ইপিনে লিখা capsulated, mostly capsulated. next, gram positive ত লিখা mesosomes are very common. Mesosome মনত আছে? The place where respiration takes place in bacteria. Mesosomes are very common. এপিনে লিখা rare. Gram negative ত rare. R-A-R-E. Rare. Next point. Remember the structure? Pili? Pili বা পিলাই জি কোৱা লিখা. Pili usually absent. Usually absent. আৰু gram negative very commonly present. Very common. Next point. Basal body has 2 rings only. Basal body has 2 rings only. What we are talking about is the flagella, right? Bracket or likha S M. Basal body ring gita ki naam asile ba S, M. Iman important point nohoy tothapi. Aru hepine likha basal body has 4 rings. Gram negative what? Basal body has 4 rings. কি কি আছে? LPSM. LPSM. Next point. Gram positive ot likha only few forms are pathogenic. Only few forms are pathogenic. PATHOGENIC. Ipine more forms are pathogenic. Pathogenic mane ki? Disease causing, right? নেক্সট লিখিবা এক্সাম্পলছ গ্ৰাম পজিটিভৰ আৰু গ্ৰাম নেগেটিভৰ examples দিছো examples not so very important right এটা গ্ৰাম পজিটিভ আৰু গ্ৰাম নেগেটিভৰ যোনখিনি পইণ্টছ লিখিছা তাৰ ভিতৰত কেইটামান পইণ্টছ মাৰ্ক কৰা এটা ফাৰ্ষ্ট পইণ্ট that we have হ'ল ন লিখা ন লিখি লোৱা জেন্থমোনাছ ভিব্ৰিঅ এটসেট্ৰা examples not important huh okay so jetia ami gram positive or gram negative or kotha ko the first point that we remember is they will retain the color they will not retain the color so first point to mark koribo na lage it's well understood right so apart from that ketaman point mark kora point number 3 single layer double layered word to cell wall single layered car thake cell wall double layered car thake right point number 3 to tick kora এটা point number 2 ne point number 3 no next asile point number 4 5 to muramic acid or point eta ase so muramic acid present or almost absent ba basically present code gram positive what right next likha ticoic acid present absent hetu point mark kora next mark kora depa point 2 next point also it's important more sensitive to penicillin less sensitive to penicillin gram positive more sensitive next likha mostly capsulated mostly non capsulated gram negative bilak ki hoy capsulated hoy next pili point 2 that's all so out of all the points that we have written those are the points that are important so etialoge ami what we have done is we have talked about the structure of the prokaryotic cell but structure of bacteria next ami ki poribo laibo life process so heading koriba life process of bacteria so egita bostu poribo libo growth respiration nutrition aru reproduction in bacteria the first heading likhiba growth ba bacterial growth likhile hobo growth in bacteria bol likhile hobo So bacteria just have an idea, right? Ekhini portion of a question nahe. So when we talk about growth of bacteria, suppose eta bacteria তুমি growth to study কৰিছা, study কৰি পেলাই যিমানখিনি information পাইছা ন, এই গোটেইখিনি information যদি আমি in the form of a graph আকো, then we'll get a shape of a, or we'll get a shape something like this, right? So প্ৰথমতে initially কমকে grow কৰিছে, তাৰপিছত বহুতকে grow কৰিছে, তাৰপিছত slow হৈছে. So this is what is called as phases of growth, right? So when we talk about phases of growth, okay, so when we are talking about growth of a bacteria, we are studying about the growth of the bacteria, and all the information that we get, we plot it in the form of a graph, or we plot it in the form of a graph, and in the graph we get a curve. The curve looks like S, so S shaped, and this curve is called as ki sigmoid curve, right? So jete ami etu shape dekha pao no, etu ami ki boli ko, sigmoid boli ko, right? Sigmoid curve. And what does the sigmoid curve say? That this talks about the growth. Now, looking at the curve, what we know is the ekhini phase of the growth of bacteria was very less, right? That's a hotate parhi gol, that's a komi gol, last of death, right? So all these four phases. So we have phases like a duta word upon bhalke sabai. We have a phase that is called as lag phase. Lag mane ki lagging behind, kom ke hoise. Next phase to lag no hoy kintu log hoy. Right? Log phase is called as ki exponential. Exponential mani ki bahut beshi hoy gol. That is called. Aur aapse lahe lahe stationary ho jabo no. Aur last of death phase. So yar pra question kiya hai je bacterial growth or phase keita saita maximum growth kuntu phase ot hoy. Log phase by exponential phase ot hoy. Slow growth, this growth constant kot thakibo. Stationary phase ot, right? And gradually it will decline, right? Aru eta question ahe, bacteria shows dash type of a growth curve. It shows a S-shaped or a sigmoid curve or growth curve. Is the point clear? Baki kini iman important nohoy. So I think that's all for today. We have started with the life process. Next class o karne respiration heading kori rakha. Thank you.